Hi guys, as promised, here is a little flow diagrammy thing covering lessons 3.1 to 3.8 for exchange systems. So this is all about the need for exchange systems all the way through from fish and insects and plants up to oxygen transport. So lesson 3.8 when we were looking at hemoglobin and the way that it moves oxygen around the body. So we're going to get started with the need for exchange services, so a brief overview. Just as a heads up, in terms of paperwork wise, this took me 4 sides of A4 to do, and it's a mess. So I would recommend having a bit more paper stuck together if you're doing it as a big mind map. I'll try and keep it neat and try to do as few links between the major points, so like fish and insects will be kept separate so that you can keep those on separate pieces of paper if you wanted to. So we're going to get started with a general overview about exchange systems, and I'm just going to start up here because it makes sense. So exchange systems, and before we begin we need to think about how and why cells need to transport substances. And this is because all cells are living, and all cells are living and therefore metabolically active. So this means that they're doing chemical reactions all the time, and in doing that they're going to be producing waste. Okay, so they're producing waste that needs to be removed, but also metabolically active organisms need nutrients, things like oxygen, things like glucose, and that's not just going to come from anywhere. So the need for transport also comes from the fact that most organisms are multicellular. So that means that they're made up of different cells that are collected to form things like tissues and organ systems. So if each of these are cells, they're all bathing in some kind of fluid, which we call, sorry, that orange highlight is awful, tissue fluid. And the tissue fluid is there to transport substances to and from cells. Now this tissue fluid is super duper important because in multicellular organisms, the central cells of the body are too far away from the surface. And that means that in most cases, diffusion is not suitable. Or not that diffusion isn't suitable in any sense, diffusion is not suitable as a transport mechanism. Okay, so now we're going to have a carry on thinking about that metabolically active or metabolism even, and make another little branch, because this links quite nicely into the multicellular unicellular thing. So the more complex the organism, so we can link that to multicellular, the more complex the organism, the higher the metabolism. And if we're not sure what metabolism means, it's the rate of chemical reactions in the organism. And I'm just going to write reactions RxNs because I'm a lazy writer and it's easier for me to do it shorthand. And because I know what I'm talking about, you can absolutely write reactions if you want to. So rate of chemical reactions in the organism. Normally we're talking about the body, but we're also going to be looking at plants later on, and I don't really think that we should call them bodies. Now, the more metabolically active, so the higher the metabolism, the greater the need for oxygen and the greater the need for removing our waste. Okay, and therefore we have to have a dedicated transport system, a dedicated exchange system. Conversely, if we are not very metabolically active, so maybe we are unicellular, made of one cell, things like prokaryotes, then they're less likely to need as much, so they don't need a dedicated transport system. So we've got a lower metabolism. And this complexity idea links nicely into the final part of this first big branch, which is the surface area to volume ratio. Sorry, I'll zoom out a little bit so you can still see over there. Surface area to volume, or I'm just going to write surface area to volume, just in case you're not too sure. So with the surface area to volume ratio, it is literally a comparison of the surface area to the volume. And we are generally writing it as a ratio. Ideally, it would be something to 1 or 1 to something. I'll write that up there. So 1 to x or x to 1. And it always goes surface area colon volume. So it's not ever volume to surface area. Now with our organisms, the less complex the organism, then we've got fewer cells making it up. Okay, things like prokaryotes. And therefore, they're going to have a higher surface area to volume ratio. Because normally the cells aren't far from the external surface. Or cells aren't far from the outside. And therefore, diffusion is a suitable transport mechanism. However, in our more complex organisms, we know that they're going to be made up of more cells. So they're going to generally be bigger, they're going to have organ systems. They've got a lower surface area to volume ratio, and they're likely to be more rounded compared to the less complex organisms, which are generally either single cells or flatworms. Or rounded, we could say 3D as well, but again, those central cells are going to be far too far from the external surface, so diffusion is not suitable. And with our surface area to volume ratio, we've got a few calculations that the examiners love to bring in because they're pains in the bums. And, you know, why not make biology even harder with some maths? So with these calculations, you have got to know how to calculate the surface area to volume ratio of a cuboid, of a cylinder, and of a sphere. Now, a cuboid is hopefully quite commonsensical. It's just a big 3D rectangle. So the surface area, and this is going to get a little bit squished up here, is 4 in brackets times the length times the height plus 2 times the height times the width. So you've got 2 faces on either end of that big rectangle, and then 4 longer, more oblongy sides. I can do a little sketch if you're not too sure. So the 2, let me highlight, that 2 height times width is for those little faces on the side. If I made that smaller, that'd probably be better. And then the 4 length times height is for the faces around the body, or the body, you know what I mean. For the volume, for cuboids, it's nice and simple, length times width times height. Now for cylinders, we start using pi and, I mean, obviously you're going to want to use a calculator. So we have got the surface area of 2 pi times the radius times the height of the cylinder plus 2 pi r squared. And if you're not too sure what a cylinder is, it's like a Pringles tube. So again, we've got the longer sides there, and that's where we get our height from. And then we have got the circular faces at the top and at the bottom, which is where we get our 2 pi r squared from. Now for volume, it's not too difficult. We are pi r squared times height. So we're using that circular face times the height just to work that out. And then finally for the sphere. Now this is the most commonly used one, and it's probably because it is the most complicated equation of them all. So we've got a big old 3D blob, and the reason they like using this is because it's very similar to what they presume a cell would be shaped like. So that radius from the centre to the outside, they say assume this cell is spherical, for example, and then you've got to use this equation. So surface area is 4 pi r squared, and then for the volume we have got 4 over 3 pi r cubed. Now for each of these, remember, surface area divided by volume, or you put it in a ratio and then cancel it down, okay? And for these calculations, the bigger the surface area to volume ratio, the better substances are able to move through diffusion. Okay, so an example of that would be if, let me put it up in a box over here. If I had 2 cells and one of them had the surface area to volume ratio 4:1 or 21:1, that 21:1 has got a higher surface area to volume ratio and is therefore, so 3 dots in a triangle is therefore better to use diffusion to transport substances. Okay, so let's just flag that up as an example. We can link that into the equation. So that is our first little section done. So now we're going to carry on. I'm going to carry on using that branch for the exchange systems because it's still sort of linked, and I'm going to go down to this bottom section of the page and we're going to have a look at the features of exchange systems. So again, this should be a nice bit of revision, but we're going to cover it all in quite a bit of detail anyway, just in case. So the first features of exchange systems, we're going to start again by looking at those unicellular organisms. And a reminder, I'm still talking about prokaryotic cells, things like bacteria, maybe some fungi, but mostly we're just going to talk about bacteria. And remember, these have got a large surface area to volume ratio, and therefore they are able to transport substances by diffusion. So diffusion is okay for moving substances. And again, to keep reiterating this point, it's because the outside of the cell is never going to be too far from the solution surrounding the cell that has got all of the substances in that we're trying to take in or get rid of. But with that diffusion, it's probably an important point to notice that even if there is a cell wall, and we know that our bacterial cells have got walls of peptidoglycans, so even if there is cell wall, we can write peptidoglycans in here, glycans. There is no barrier to diffusion. So diffusion is still going to happen regardless of whether there's a cell wall there or not. Now for other organisms, I'm going to just slide this down a little bit when we're talking about multicellular organisms. So this is anything basically more complex than a unicellular organism. We know that we're going to need transport and exchange systems. So we've known that for this assessment you are looking at gills, you are looking at tracheae and spiracles, you are looking at the lungs, and you are looking at plants as well, and that passive movement of gases into and out of the spongy mesophyll. So all of these transport systems have got common features that are seen throughout all of them, and we're just going to say they're adaptations of the exchange surface, and we're going to start by looking at how the surface area is maximised. So max surface area, and we do this in a few ways. So we can either have something called rugae, which are infoldings of the membrane. So rather than our small intestine being just a tube like this, actually, if I just rub that out, we have got lots and lots of folds in the membrane, which are going to increase the surface area for substances to move down and to be absorbed through. We can also increase our surface area through the use of villi and microvilli, and we know that they are extensions of the plasma membrane. So a cell that is vilified would be something like that, and they make up a nice little border all the way along our cells. Now what they're going to do is, again, they're going to increase the surface area and they're going to increase the space available for diffusion. So that means that it's going to increase the diffusion speed or the rate of diffusion. Similarly, most of our exchange surfaces or our transport surfaces have got a thin membrane. So if you think back to the alveoli, if you think about the small intestinal walls, there's going to be generally only one cell separating a medium, so a transport medium, things like blood, and the location of the substance that we're trying to take in. So only one cell separating regions, and therefore we are making sure that the diffusion distance is reduced. And that in turn is going to increase our rate of diffusion. If you think about it like running across motorways or roads, if I only run across one lane, that's going to be a lot quicker for me to get through to the other side of the road than if I have to run across 8 lanes of traffic. Just because there's a further distance to cover. It's exactly the same as particles. Particles don't want to have to go a long way. They've got only a certain amount of kinetic energy in them. Having a thin membrane means that we're going to just be able to get from A to B a little bit quicker. As well as this, the membranes are normally selectively permeable. Notice how I'm not writing semi-permeable. Semi-permeable implies that it's half permeable. Selectively permeable means that only certain substances can come through. And that's going to be controlled by everything that we learnt about in Module 2. So we're going to be using our channel proteins, or we're going to be using our carrier proteins. Okay, so if the substance that we need to transport isn't there, those channels are not going to be open. And what that means is that we prevent mass substance flow. And that's going to stop our cell being damaged by things like osmotic stress. So if we move too much into or out of a cell, then it might affect the water potential, and then we're going to have either swelling and lysis, or maybe flaccidity and not very good cell structure. So we can just say that we're preventing osmotic stress. And the final adaptation that these exchange surfaces normally have is a way to maintain the diffusion gradient. So that means to say that after I've moved my substance from A to B, so substance, maybe oxygen, from A to B, that substance has to be moved away, because we're continuing to ensure that there's a difference between places A and places B, so we can keep moving substances along that transport gradient. Now that diffusion gradient normally requires a medium. And a medium is just a material that holds things. But if we're saying transport medium, we're talking about a material that moves things. So we can think about that in terms of the circulatory system. We've got blood in humans and other mammals, but then we've also got something called hemocool in insects, and we haven't learnt about that just yet. That's coming though, that is in one of the lessons when we start looking at the circulatory system. And then we have got also the value of air. So air and the wind ensures that we can move substances away from the surfaces of leaves, but also away from the spiracle openings. So we can see there that we've got quite a few adaptations in those exchange systems, things like that. And we can see using Fick's equation, so if I scroll down here and put Fick's equation, that all of these different factors help to increase the rate of diffusion. So rate of diffusion is proportional to the surface area of the transport system or the exchange surface times the concentration difference from A to B, so where A and B are two separate places, divided by the diffusion distance. And you can see there that each of those factors that we've tried to limit with our adaptations are included in our equation. So they're all equally as important. So what we should see now is we've got quite a big introduction to exchange systems. If you're not too sure, I'm going to leave this on for a second. Let me know and we can go over things together. Otherwise, we're going to get straight into it and we're going to start looking at insects. I'm going to carry on working down in this left-hand side just so we can keep it neat over one side for now. And yeah, hopefully this should be helpful. If you're not finding it useful, you're more than welcome to switch it off and do your own mind map. But at the end of it, I'll be able to zoom out a little bit like this and we'll be able to see how much detail you guys have actually covered over the past 8 lessons or so. So let's dive into it then. So I'm going to use a nice navy blue now. And I'm going to flag that up as a new point using a little bubble. So we'll start with a diagram because I think that's quite valuable in terms of remembering what the structure is like. So if this is the body of an insect, so the abdominal wall, we have got the pore where the gases can enter, so gas here, and it's surrounded or covered by spiracles, and they're effectively like valves that open and close to let gases in and out. Following on from the spiracles, we have got the tracheae, a bit like the trachea in humans, which then deviates into the tracheoles. And they meet straight into the cells of the body. So let me zoom in a little bit, make it neater. Meet body cells. And in meeting the body cells, we are going to obviously reduce the diffusion distance because our substances don't need to be able to move a long way. So if we go straight from the end of the tracheoles into the cell, we're minimising the random movement of molecules. We're sort of giving them a little bit more of a direction, and that's going to increase the rate of diffusion. Another important fact for later on is that the tracheal ends are covered in fluid. Something like tissue fluid, but in insects it's probably not called tissue fluid. A few more adaptations that we've got is that in the tracheae, they are supported by rings. Okay, so they're supported by strengthened rings, which holds them open, and it means that they can stay open even if the pressure changes too much. If they weren't strengthened, they might collapse, and then obviously the insect is going to have no hope of gas exchange. So when we're talking about gas exchange in our insects, what we're really looking at is how oxygen and carbon dioxide are able to passively really move from areas of high concentration to low concentration. So in our insects, we are taking gases in and out of the spiracles. So I'm just going to link those together. Gas exchange. Okay, and remember, we're still using diffusion, so we're going from areas of high to low concentration. And it's a passive process. We're going to have a look in a second about how it could be an active process, but generally speaking, unless we're doing like really active things, maybe flying long distances, for example, it's going to be super duper passive. So we've got highest oxygen, so I'm just going to put a big O2 with an arrow going upwards for highest in the air. And we're going to have lowest oxygen, so low oxygen in the respiring tissues. And what we can see there is that between the outside air and the respiring tissues, there is a concentration gradient that I'm just going to label here. And that concentration gradient means that oxygen is drawn in by diffusion through the spiracles, into the tracheae, into the tracheoles, and then into the cells. The same but opposite happens for carbon dioxide. So carbon dioxide is highest, so highest concentration of CO2 in the respiring tissues, because obviously that's going to come from respiration, which our respiring tissues are unsurprisingly doing. Oh, respiring? No, respiration. There we go, fixed that. Obviously can't see that there's been a mistake there. And that highest carbon dioxide concentration in the respiring tissues is compared to the super comparatively low concentration of CO2 in the air. And again, we've got that downwards concentration gradient, same as last time, but the carbon dioxide moves out of the insects. And again, that is through the spiracles, or actually for the carbon dioxide, we're going from the cells to the tracheoles to the tracheae out of the spiracles. Okay, now we've got other ways that we can emphasise this gas exchange system. And so we're going to have a look at those now. So the first one is body movements. And what we mean by body movements is we're using the fact that, sorry, this is going to be an awful diagram of an insect. So we've got the abdomen at the back is made up of segments. And on each of those, there are the spiracles. But each of those segments, so the spiracles, sorry, are those little dots, those segments are able to slide over each other. So we can say that they're contractile. And so when the insect is resting after doing lots of flying, for example, so in high demand situations, we've got firstly the contraction of the abdomen, so contraction of abdomen, and by that I mean that it moves inwards. So if that's the abdomen of the insect, it's going to sort of squeeze together. What that's going to do is increase the pressure by decreasing the volume of things like the trachea and the tracheoles. And that in turn is going to force waste out. Things like, like carbon dioxide out. A bit like when you like pump sort of an accordion or anything that squeezes, You force the air out of it, a balloon, for example. But then when the abdomen extends, and notice I'm saying extension or extending rather than expanding, because it's not getting fatter, it's just getting longer. So those segments separate a little bit more. Then what we get is a drop in pressure because the volume of the abdomen has increased. And that in turn is going to result in air being drawn in. And obviously, if air is drawn in, then we're going to increase the amount of oxygen taken in as well. Which is going to be helpful because then we can get rid of the waste, but we can also take in oxygen to replenish the stores in the cells. We've also got to be aware of the fact that the ends of the tracheoles are covered in liquid, in a watery solution. So we'll do that as our point 2. Okay, so we know that the tracheoles end at the respiring cells. Okay, so I'm just going to do this as like a little step by step. So at the respiring cells, if they're in low oxygen conditions, we call that a hypoxic condition. And that's a new keyword that you can use if you want. So low oxygen, then we're going to respire anaerobically. And what that's going to do, as we know, is produce lactate or lactic acid. And that is actually going to dissolve in the cytoplasm. We're talking about the cytoplasm of our respiring cells there. Now, when that dissolves in the cytoplasm, what's actually going to happen is that it's going to reduce the water potential of the cytoplasm because we've got more dissolved stuff in it. And therefore we've got a water potential gradient between the ends of the tracheoles. Oh, there's a dividing part there. Ends of tracheoles and the respiring cells. Now what that means is that water moves from the ends of the tracheoles into the cells. And again, that's going to result in our, what comes next? EF. That's going to result in our favourite thing. So we're going to drop pressure by increasing the volume available in those tracheoles. And that's going to ultimately increase the air and the oxygen drawn in. Okay, so as you can see, in combination of those body movements and the fact that the end of the tracheoles are covered in water, in highly metabolic situations, so when we are respiring anaerobically, if we're an insect, we're going to get active movement now of oxygen in. So we're working to drop the pressure and increase the volume available in those tracheoles, the tracheae, and try and draw as much air and therefore oxygen in. The important thing to know, I'm just going to chuck back up to these spiracles bits again, up here, is that the spiracles are important because they can open and close. So this is the last little bit that we're going to do on the insects. And the spiracles are sort of a limitation of the gas exchange system in the insects. So the limitations of the spiracles. So if they're open, then we can have gas exchange, but also we can increase the chance of dehydration because water is going to be lost at the same time. And likewise, if they're closed, then there's going to be no gas exchange or little gas exchange, but they're going to reduce the chance of dehydration. So the spiracles are there fantastically as sort of like an opening closing thing, and because they're sort of a double-edged sword, they can be both beneficial and a curse. They're only opened at selected times. And that's why the insects rely quite a lot on these body movements that we've spoken about and the fact that the ends of the tracheoles are covered in water. Normally, they're going to be closed to limit the water loss. So the passive movement of oxygen and carbon dioxide in and out of the insect body is not necessarily going to provide enough oxygen. So let me zoom out there a little bit. You can pause now if you need the information. I'm sorry about my horrible writing. And then we're going to look at fish. So as you can see so far, we've got loads of detail. And I hope you find this really useful. It's exciting to see how much knowledge you've actually gained yourself. So I'm going to pick a lovely green colour now. And we're going to move on to fish. I'm going to move down into this corner so I've got a little bit more space. So fish, nice big bubble. And we know that in fish, if I do a horrible diagram again, because that's my favourite thing about these little videos, I've got big bubble lips. Oh, you know what? That's not too bad, a fish. All of the fish that we're talking about, we're not talking about flatfish. We're talking about the general fish that have got barrel-shaped bodies. Ooh, barrel, if I could spell. And what that means with that barrel-shaped body is that we've got a low surface area to volume ratio. And therefore, diffusion is inefficient. Because of things like that surface area to volume ratio being quite low, but also the fact that the scales are waterproof. So there's no way to get oxygen-containing water near the body tissues anyway. So instead, we're relying on, if I put a little eye there, gills. Okay, and the gills are, I'm just going to link that to the main heading. The gills are our transport and exchange surface. And they're made up of lots of different layers to maximise the surface area of the exchange surface. So we've got a gill plate, and I'll label these as I go. So gill plate that's got gill filaments sticking out of it. You can see it looks a little bit like an ugly toothbrush. And those filaments, if we do a cool zooming in kind of image, if we look at those filaments side on, like that, they've got something called lamellae on them. Okay, and the gill lamellae are perpendicular, so L are perpendicular or at 90 degrees to the filaments. And all that's going to do is that's going to maximise our surface area for oxygen absorption. And ultimately, we're looking to get as much oxygen out of that water as possible, because we know that water is just passing over the gills. So the more space we've got available for those, or for that oxygen absorption, the more efficient the whole process is going to be. So now we need to look at exactly how those gills are going to take in the oxygen and how oxygen even gets there in the first place. So, step one, the fish is going to open its mouth. And what that's going to do is that's going to increase the volume of what we call the buccal cavity or the buccal cavity. I don't know how to say it. It's its mouth hole. And in increasing the volume, that's going to decrease the pressure because they are inversely related, and therefore water rushes in. Okay, so water is going to be drawn into the fish's mouth. Step two, though, the fish closes the mouth. And what that's doing is it's going to decrease the volume and increase the pressure, which is going to force the water elsewhere. And now that elsewhere is actually backwards. So step 3, water is forced over the gills and then is able to leave the body. But as water is forced over the gills, so we'll do a little underliney thing, forced over the gills, that's where we get our oxygen absorption. And if you remember from the lessons, that oxygen is absorbed through countercurrent flow. And this is our final little bit of fishness. So countercurrent flow is where the water and the blood flow in opposite directions. And when we're talking about blood flow, we're talking about the blood that's found in the gill lamellae. So as the water rushes over the gill lamellae, the blood flow going in the opposite direction is going to ultimately result in a constant concentration gradient. So diagram wise, we've got something that sort of looks, let me choose some different colours. Hold on, let me pause and I'll come back with a diagram. Ta-da! As if by magic. So there we've got a really ugly diagram, but what we can see is that the blood flow is going in the opposite direction to the water. Now what that means is that every stage of the blood circulation, so all blood, is meeting water with a higher concentration of oxygen, and therefore we've got a constant diffusion gradient. Okay, so at the very beginning of the water passing over the gills, we can see that the water is 100% saturated, so I'm just looking here. The water is 100% saturated with oxygen, and it meets blood that's 80% saturated with oxygen. So we're going to get diffusion from high to low concentration, and again, that's going to happen all the way down. Even when the water is no longer particularly oxygenated at the end of the lamellae, it's still got more oxygen in it than the blood. And in a graph, we've seen this graph before, we've got the y-axis with oxygen concentration, and we've got the x-axis with distance along the lamellae. Our graph always has concentration of oxygen in the water above the concentration of oxygen in the blood. Okay, so blood, and then we've got the water just there. And in an exam situation, it might ask, what is the benefit of this? It's the fact that we've got that constant diffusion gradient. So I'll put a little note at the end, these green arrows showing us constant gradient across the whole length of the lamellae. So all the way down. And actually at the end, so we can put a little mark here, it might show it on the graph itself, almost 80% of the oxygen in the water is absorbed. So the water might go from 100% saturation down to 20%, and therefore that 80% of oxygen has got to go somewhere. So luckily, the fish stuff isn't too detailed, it isn't too complex compared to the insect, I don't think. We've got a couple more organisms to look at. We're going to go for plants, and then we're going to go for humans. I recognise that this video is a lot longer already than I was hoping it was going to be. I'm sorry. But again, if we zoom out, there's so much stuff there. Okay, so we're going to keep pushing on. We're doing fab. Proud of you for sticking it out this far if you've managed to watch the entirety of me mumbling. So yeah, let's get started on the plant stuff. So we're going to go for a lovely lush orange for the plants. I'm just going to zoom to the top because this is quite a big bit and I want to leave maximum space for the human stuff. So let's get started. Little plants with a cute little bubble. And we're going to start by looking very briefly at the anatomy of a leaf, because that's what this whole gas exchange system is related to. So we've got the upper and lower layers that are made of a very waxy substance, a waxy cell type, and we just call it the waxy cuticle. And it's there, it is waterproof, and it's a protective mechanism. And what it can do is produce chemicals in there that are going to sort of heal the plant if it ever gets damaged. Just underneath that, we have got the palisade mesophyll layer. So lots of upright cells that are all, we put the nucleus in there. The palisade cells, which are all specialised for photosynthesis. So they've got lots of chloroplasts. And I know this is not entirely relevant to gas exchange, but it will be in a second, because then we've got the spongy mesophyll layer. And these are cells which aren't as tightly compacted. So I'm going to put the labels over to the left-hand side. Spongy mesophyll. And those spongy mesophyll cells have got air spaces between them. And what this airspace means is that we've got free movement of gases. And then at the bottom, we have got another waxy cuticle layer. I'm only going to draw it small this time. But we've also got guard cells. Okay? And I know they look a little bit like my awful diagrams in the lesson of channels, but they're effectively channels anyway. So guard cells are another type of specialised cells that we're going to have a look at later on. And they are causing the opening and closing of the stomata. So the stomata are those pores on the underside of the leaf, and they're going to allow gases into and out of our cell. Now, the important thing that we need to know about plants is that gas movement is passive. What that means is that we are relying on the natural kinetic energy of the gas molecules themselves. And because of that, we can't control how fast or how slow gases move in and out. We can control water loss, which we'll look at later on, but we can't go like, okay, breathe in plant, breathe out plant. It doesn't work like that. However, that gas movement being passive doesn't alter the fact that the metabolism of plants is variable. And by variable, I mean it can change, and that change is dependent on the need of the plant. So if it's in a growth phase, maybe it's going to want to do lots of respiration to get loads of sugars so it can lay down some more organic molecules, maybe grow some fruits. Maybe, though, it just wants to photosynthesize and develop some more glucose in that way. Okay, so we've got our structure. So let's take a look in a little bit more detail at how the gases are exchanged. I'm going to move over here now. And the gas exchange, we've already said, relies on the stomata and them opening or closing. And that stomata opening or closing is controlled by the guard cells. And the guard cells have got lots of different adaptations. But these stomatal opening and closing, very similar to the spiracles on the insect, is a double-edged sword. And this is going to bring us to the next bit of the plant stuff later on. Because, again, if we've got the stomata open, then we can have gas exchange, but also water loss. And if they're closed, then we don't have gas exchange, but we don't lose water. And for this reason, the guard cells are really, really useful. So what they're going to do is they are going to open when water is in full supply. or when water is available. Okay, and if they open when water is available, it sort of nullifies the problem with losing water when they're open. So gases can be exchanged. And this is because the guard cells are turgid. And if we draw that in a little diagram, that means that they've become swollen and sort of bean-shaped because they've got a thicker wall on the inside of the cell. We'll just label that in there. Thick wall on the inner layer, and that means that the pore is open. However, they are closed when water is unavailable. Or when water is a stress, so we can say water stressor as well. And what this means is that our cells are now flaccid. It means they don't have enough water in them, they're a little bit floppy. The vacuole isn't as swollen as it could be. And ultimately, that's going to close my pore. So it's sort of a protective mechanism. If there's enough water, then the plants are like, hell yeah, let's go and exchange some gases. If there's not enough water, hell no, we are not going to exchange gases. We don't have time for that. We don't have enough water to be opening and dehydrating ourselves. But some plants that live in dry conditions anyway are still able to exchange gases. They're still going to need to exchange gases. And those plants are called xerophytes. So we're going to go from this water stressor down into xerophytes. Okay, and these are plants that are able to survive when water is in short supply. And by short supply, we're talking about deserts, because obviously they're quite dry, but also icy regions. And icy regions could be things like the Arctic, because that water is not available. It's locked into the ice structure. By survival as well, we mean that they're able to reproduce. They're able to grow, not just survive. Because, you know, living is not just about surviving, it's about growing and reproducing and actually adding to the ecosystem. These xerophytes have got some amazing adaptations to help them survive, and that's what we're going to look at for this final little bit of the plant stuff. So we're going to take each of them in turn. We're going to have a look at why they're important, maybe draw some diagrams and things like that. And the first couple sort of go hand in hand, and those are a thick waxy cuticle, rather than just being one cell thick, and also some sunken stomata. And the reason they go hand in hand is because the guard cells can't really change their size or their thickness. So in terms of the leaf surface, they might look a little bit like this. Okay, so those waxy cuticle layers at the top are much thicker than the guard cells. So sorry, the air is going to be moving in following that arrow. So the guard cells are sort of set back from the surface. And what this means is that if I draw a little purple arrow, if air rushes across the surface of the leaf or the underside of the leaf, it's not going to disturb the air in the guard cell or around the stomata. So we can say there that we're developing a microclimate, where that big purple squiggle is, of humid air. And humid is just to do with the fact that there's an increased water concentration in that air, which is going to reduce the water potential gradient between the inside and outside of the cell, or between the inside and outside of the leaf. And what this does is it enables the stomata to open so we can exchange gases, but reduce the water loss, because there's not as much difference between the two areas. So if you think about the outside air and the inside air, so we might have in the spongy mesophyll that concentration of water, but in the outside air, because of that microclimate, there might only be a very shallow gradient. So water loss isn't really happening. Another adaptation is the fact that these xerophytes normally have reduced numbers of stomata as well. And this is going to help because it's just going to mean that there's less pores for water to leave by. So reduce water loss points or pores. And in turn, that is going to be a little bit problematic because we're going to reduce the possibility of gas exchange. Not fab, but sometimes it's a sacrifice that we've got to make. We'll carry on up in this top corner looking at the leaves, and there's quite a few adaptations that they've got. So let's just zoom up here and make a nice little subheading for the leaves. So when we're looking at the leaves, we're going to look at the reduced number of leaves. We're going to look at the fact that some of them are curled, and also the fact that some of them are quite hairy. So let's start with the reduced number of leaves then. Obviously, if we've got fewer leaves, then we're going to have fewer stomata. But actually, by reduced number of leaves, I mean that the leaves are more likely to be spines in terms of things like cacti, or needles in terms of things like fir trees and things like that. Now, in having these spines or needles, what we're actually doing is we're making the leaves slightly more circular. And in doing that, they've got a smaller surface area to volume ratio. In the hairy leaves, what we're doing is we're providing a layer of insulation. So if this bottom horizontal line is the leaf surface, and those hairs are upright, sticking perpendicular, then the air, much like that sunken stomatal pit, the air gets trapped in a little layer like a duvet. And again, like those sunken stomata, we get a microclimate. And that microclimate is of humid air. And it just collects in there, it holds it, and it's going to reduce the water potential gradient between the inside and the outside of the leaf, which is fantastic because it's going to minimise water loss while enabling the exchange of gases into and out of the plant. Now the final one is those curled leaves, and this is normally used in conjunction with the hairy leaves. So I'm going to link those together actually. So in conjunction with other adaptations. And what we're doing is wrapping the leaf in on itself so it forms like a nice horseshoe shape. And again, we're focusing on that microclimate. So that stagnant, humid air that can't be moved away. The wind can't get to it because effectively the curls around the leaf, or the leaf curling in on itself, stops any wind being able to get in there. Okay, and therefore we are reducing the water potential gradient again, so limiting our water loss. And that is all of the stuff about plants done. So I'll zoom out just so you can see all of the plant stuff. It's not as visible as the other ones. I'm sorry, that orange pen wasn't ideal. But the final section that we're going to move on to for this last little bit is all about humans. So hopefully this shouldn't take too long. I imagine maybe another 15 minutes. But again, if you feel confident in it, you're welcome to skim and zoom through. That's absolutely fine. So let's get started then. And we're going to start, I'm going to use this nice hot pink colour. I'm going to start by looking at the need for exchange systems. So this pink stuff is all the human. And the need for exchange systems in humans is really because we are very complex. And in being complex, we are highly metabolic. And what this means is that we need lots of oxygen, and we also produce lots of carbon dioxide. Okay, so that oxygen needs to be taken in, and that carbon dioxide produced needs to be taken out. In terms of highly metabolic, we're also referring to things like the production of ATP, our energy molecule, and that's produced through respiration, for which oxygen is key. So you can see that we're starting to link things together. Now, if we don't lose or get rid of that carbon dioxide, it can start to become toxic. It can start making our blood really acidic. That's problematic because it's going to start damaging things like our enzymes. Now, with our need as well, we've got to think about the need for internal organs. Okay, why are our lungs on the inside? Why don't we have spiracles? And the reason we've got lungs on the inside is because they are protected. And they're protected by things like the ribs and the sternum. The sternum is your breastbone. You can feel it if you feel sort of between your cleavage, I guess. It's also there to prevent dehydration. So the wind can't blow against the lungs and dehydrate them from the surface. So we're not having as much water loss. Finally, having those organs internally means that they can be supported. So we've got things like tissues called mesentery, which sort of support the lungs. So they're not just sitting there, they're held and supported by other tissues and prevent collapse. Now we're thinking about, firstly, the organs of the respiratory system. And I'm going to do an ugly diagram because I find it most useful, and then I'm going to label it afterwards. So you guys are welcome to draw. You're welcome to ignore. I find the drawing quite useful. Obviously the alveoli, the little mushroomy things, would be on both sides, but I'm only going to draw them on one. So we start up with a general overview of the lungs. Okay, and the lungs are lobed. That means they're made of a couple of interconnecting parts, and we know that they're protected by the ribs, and the oxygen that the lungs take in has to be supplied from somewhere. Now obviously that's in the outside air, and that air first starts coming through into the lungs through the trachea. Okay? Now the trachea is adapted to this high volume of gas passing through it by the fact that it's got something called the D rings. And these are cartilage rings, so we can say that the trachea is cartilaginous, and that's there to support the trachea opening. And if you're not sure what I mean by support, if you feel on your neck just now, you should be able to feel the vibrations of you going like, ah, for example, but you should also be able to feel some little ridges. And those ridges are those D rings of cartilage. In the trachea, we've got some protective mechanisms as well. So we've got not only the D rings to help support, we've got ciliated cells and we've got goblet cells. And the ciliated cells are similar to villi, so they've got those little layers on the top. And the goblet cells are there to produce mucus. And the cilia and the goblet cells work together to form part of the primary immune response, that non-specific immune response. And we learnt about that in module 2. Okay. And that trachea then deviates down into these two tubes, which I'm going to link together and put over here, called the bronchi. And the bronchi, as I've said, we've got two of them. They are also cartilage containing. So they're held open. But as they move towards the bronchioles, so as they get smaller, the amount of cartilage decreases. In their walls, they are made of muscles, so they are slightly contractile. But once we get into the bronchioles, which I'm going to move to now, they are further deviations of the bronchi. And these are not cartilage containing. Instead, they are held open by smooth muscle. So they've got moderately thick walls. But as we get even smaller going towards the alveoli, they start to get even thinner and thinner. And ultimately, we're going to reduce the diffusion distance by making our walls thinner. The final part of the lungs, the respiratory system in this case, is these little mushroomy things called the alveoli. And these are lined with epithelial cells, which, as we know, are really, really thin. Going to reduce the diffusion distance again. But also they are highly vascularized. And that means they've got a massive blood supply. So vascular, high blood. And that blood supply is our transport medium. So we're moving our oxygen away and we're maintaining the diffusion gradient. Okay, so now we know a bit about the structure of the respiratory system. Let's have a look at how we actually breathe. Okay, and breathing, we know, I'm going to just put over here, is not called breathing now that we're A-level students, we're calling it ventilation. And the whole idea about ventilation is that we are trying to get oxygen in and carbon dioxide out. Okay, so we've got two different stages of ventilation, and the first is obviously breathing in, or inhalation. And this is an active process. It's going to require energy. Where do we get that energy from? We get it from ATP, which is made during respiration. Now for inhalation, there are a few steps that we need to go through. And the first is that the diaphragm, spelt diaphragm, contracts. And that means it becomes flat. And the external intercostal muscles also contract. Okay, so this is going to result in the ribs moving up and out, and it's going to increase the volume, decrease the pressure of the thorax. That's our chest cavity. And in doing this, air rushes in to equalise the pressure. And obviously, if air comes in, oxygen is supplied. For exhalation or breathing out, the opposite sort of happens. So exhalation. This is passive, much more related to just recoil or relaxing of muscles. So step A, the diaphragm, and again, diaphragm relaxes, and the internal, and I'm going to write IC for intercostal because I don't have a lot of space here, internal intercostal muscles contract, and in doing this, the ribs move down and in. This is going to result in a reduced volume and increased pressure in the thorax, and therefore the air is forced out. Now, the important thing to recognise with this ventilation, we've said about lots of different muscles. The muscles are generally only used, especially in exhalation, under periods of massive stress. So after high activity. So if you've run for a bus, for example, and you're absolutely puffing by the time you get on the bus, this is what you're going to be doing. Normally, we're just using elastic recoil to move our gases. Okay. A few other things related to ventilation that are important before we look at the transport of oxygen is a couple of equations, and we know it's super annoying when biology mixes with maths, but here we are. So we've got the first equation, the pulmonary ventilation rate, or PVR. I'll just write what that stands for up there. And that's measured in decimeters cubed per minute. And that's equivalent to the tidal volume times the breathing rate. And if you're not sure what the tidal volume is, we'll just go over that quickly. It is the volume of air breathed in per breath at rest. And rest means that you are inactive, you're laying down, you're at a standard temperature and pressure of the atmosphere. And that's measured in decimeter cubed. And obviously breathing rate is just how many breaths you take per minute. Okay. We've also got to consider as well the FEV. Oh, didn't want that pen. Let me go back. The FEV. And the FEV stands for the forced expiratory volume. And the reason I've put this in calculations is not because there's an equation to link with it, but more because it can be used in calculation questions when looking at how diseases have impacted breathing. So the FEV is actually the maximum volume of air breathed out in 1 second. Okay, and diseases such as asthma or pulmonary fibrosis, where you've got like little tumory kind of things on the lungs, little fibroids, are going to result in a reduced FEV. And oftentimes, these exam questions will ask why that's going to result in fatigue. And fatigue comes because oxygen cannot be carried as efficiently, because it's not transported in as well. We've just got a lack of oxygen. And if we haven't got enough oxygen, then we've got a reduced contraction of muscles. They're not going to be able to contract as well. So reduced contractions. And we've got reduced ATP formation. Now, obviously, you might have seen by the time I lied at how long this was going to take, because as you can see, we've covered quite a lot in pink already. We have just got one little bit left to go through, and that is all about hemoglobin. And I promise, then it's nearly done. But look at the detail, guys. I mean, if you've managed to keep up so far, you should be absolutely chuffed with yourself. So the final bit that we're going to do, I'm just going to shoot straight up here, and we're going to link it to oxygen supply up here, is hemoglobin. And we can abbreviate hemoglobin to big H, little b. Hemoglobin and oxygen transport. So the first thing we're going to look at is the hemoglobin structure. And in terms of structure, it is a tetrameric or it's a tetramer. So it's got 4 different subunits, and it is a globular protein. By tetramer, I know that that means that it's got 4 chains, as I've said. So it's got 2 lots of alpha chains and 2 lots of beta chains. And each of those chains has got a heme subunit, which it contains an Fe2 plus ion. Remember, in an exam you can put ion, so ion like that, or Fe2 plus to carry the oxygen. Okay. In terms of hemoglobin being able to carry the oxygen, we need to know that there's that reversible equation. So hemoglobin plus 4 lots of oxygen, because each heme unit can carry an oxygen molecule, not an atom. It's a reversible reaction to form this oxyhemoglobin. Okay. So this oxyhemoglobin, obviously, we can see is a reversible reaction. And it's reversible because oxygen can either associate or dissociate with the hemoglobin, depending on where we are. So we're going to add a little bit here, oxygen loading or unloading. Okay. So oxygen loads at the lungs, because obviously there's a higher concentration of oxygen there, and it unloads at the respiratory tissues or the respiring tissues, wherever that may be. And then we're going to have a look at how we can link affinity into that. So at the lungs, oxygen has got, or hemoglobin, sorry, has got a high affinity for oxygen. Now this means, and I'm just going to come down here now, oxygen, O2, binds to hemoglobin easily. So it wants to stick to it, but it's hard to let go. However, for unloading, hemoglobin has a low affinity for O2. And this means that O2 doesn't bind easily, but is let go easily. All right. Now, we know, because we've done this lesson already, we've done this as part of a topic already, that the affinity for hemoglobin to oxygen also depends on the partial pressure of oxygen, or PO2. And that's just to do with how much oxygen there is in a region. So the higher the PO2, or the partial pressure of oxygen, that means that there's more oxygen available. And we can see this, this relationship between oxygen affinity and the partial pressure of oxygen using the oxygen association curve. So as we know, we've seen this before, we're just going to quickly label it up. Wow, that is a horrible x-axis, I'm sorry. On the x, we're going to have the partial pressure of oxygen, remember that amount of oxygen, and up the side, we're going to have percent saturation. Now I know that hemoglobin can only carry 4 oxygen molecules at a time, so I'm just going to split the percentages up into 25, so 100% would imply that all 4 heme groups are occupied, 75% means that we're 3/4, and then 50%, 2 out of 4 of the hemoglobin subunits or the heme subunits occupied, and then 25%, only 1/4 are. And we should know and be confident in drawing the fact that our graph is very shallow to begin with, and then there's a rapid upshot, and then it flattens off. And there are many reasons for this, But we're going to have a look at positive cooperativity, and that's the relationship between the oxygen binding and the structure of hemoglobin. So we'll start down here. At low partial pressures of oxygen, hemoglobin has a closed structure. Okay, so none of the subunits, or the subunits are very unavailable. It's nice and closed. But eventually, 1 out of 4 of those hemoglobin subunits, so 1 out of 4 Fe2 plus, bind oxygen. So now we formed that first step towards the fully saturated oxygen, or oxyhemoglobin, sorry. And that's going to start opening up our hemoglobin structure. And that means that we're going to increase the availability of the binding sites. Okay, now as my partial pressure increases only a little bit, we get a rapid binding of more oxygen. And this is because that hemoglobin becomes more open in structure, but also there's more oxygen available. So more O2 available, and more open hemoglobin. And by this point, at 75% saturation, we are looking at a molecule like that. Okay, but after this 75% saturation, you can see that the graph starts to plateau. And the graph plateaus with increasing partial pressure of oxygen because, just due to chance, it's very unlikely that the 1 out of 4 heme groups left is going to be able to bind to that oxygen. It's not impossible, that's why it does reach 100 eventually, but it's unlikely. Okay. Now, when we're seeing this graph, we also know that different species are going to have different shapes and locations of the curve on the graph. So we're just going to add a couple of annotations. If, and I'm going to put it up here, if the curve shifts left, the hemoglobin has a higher affinity for oxygen. And remember, high affinity means that it loves it, it wants to hold on. Okay, and these would be organisms that live in low oxygen conditions. Maybe things like flatworms that live in the sand. However, if my graph shifts, and I'm just going to put this in here, to the right, and I'll label it down the bottom so you can still see what I'm doing. This graph moves to the right. The hemoglobin has a reduced affinity for oxygen. Okay, and that means that it's going to not want to hold on to it, and it's going to let it go easily. Now, to finish up, we see this shift to the right in high carbon dioxide conditions. So when the PCO2, the partial pressure of carbon dioxide, is high, we shift the affinity of hemoglobin for oxygen to the right. And this is what happens at the respiring tissue. So the graph shifts to the right because the carbon dioxide has a negative effect on the hemoglobin structure. Okay, because the carbon dioxide is able to dissolve in solution to form carbonic acid, and that's going to affect the 3D structure of our protein. And this shifting of the affinity is called the Bohr shift, relating to the properties of carbon dioxide. Now, as you can see, there is lots here. Okay, I'm just going to zoom in on this little bit just for a second, just so you can see all of that hemoglobin and oxygen transport stuff. I appreciate that there is a lot to take in, and I want to thank you for getting to this point. If you did, for sticking with it, I hope you found this useful. If we use this in conjunction with some exam questions that I can help you with, then we're going to be absolutely golden. Now, the next one I make of these is probably going to include things like the heart and plants, which is potentially more interesting than fish and insects. And yeah, I'll see you again next time. Bye!